Yes, my pleasure to introduce Yuqi Tan today. She's a postdoc from the Nolan Lab at Stanford, and she's going to talk about, changing the title, Images to Insights, Spatial Biologist Guide to Decode Multiplex Image Analysis. And just briefly about her bio, she holds a bachelor in cellular molecular biology from the Chinese University of Hong Kong and PhD in computational biology from Hopkins, and she has numerous scholarships and awards, many to mention, and in particular specializes in crafting computational tools for integrating different types of spatial data sets. And yeah, looking forward to learning about your tricks and all the advice on how to tackle spatial data sets.
Thank you so much for the invitation and the kind introduction. Thank you guys so much for coming in pre-holiday. A little bit more thanks for the kind introduction. So I am a computational experiment hybrid. So started here as a cell molecular biologist. And during my undergrad time, I travel a little bit around different continents and also do a little bit experiments and in different parts of the world, working on model organism from plants all the way to mouse. But since my PhD, I switched primarily to computational method development, working with Dr. Patrick Cahan at Johns Hopkins University. And now I'm here with at Stanford with Dr. Gary Nolan, working on spatial omics method development as well as technology development. So I have always fascinated by images. So whether that's in the form of single-cell omics, where you have the embedded form of all the data that you can see in a picture form, or in the spatial omics field where you read out is beautiful images from the sample that you have selected. I've always also fascinated by 3D images where you can see your tissue's architecture in a non-interrupted Z-stacked. However, my primary interest is using machine learning to integrate and also look at all of these different data modality and extract insight from them. So today I will just pick a vignette of my projects, which we call SPAR Codex or SPACY for short, because my lab, we love space. And SPAR Codex is because we really want to massage the Codex data to a manageable form. And this is the spatial data analysis package that we hope to put forth, and it's in Python. So this audience probably needs no introduction as to how we can use spatial data to interrogate the tissue or tumor microenvironment to know how the cells interact in its native environment. And many people have asked me the question, there are so many spatial platforms, which one is the best and how do you tackle them, right? So having said that, if you actually look at all these spatial data, they can all sort of massage into this common output feature where you have X, Y, or Z coordinate, and then some sort of marker features. And so for today, even though I will be primarily using Codex as an example, trying to tell you how we handle Codex data in the lab and hopefully helping you to understand it, just bear in mind some of these technology that you might be working on, which might not be Codex, could also utilize the platform in a shape or form. So for people who are not as familiar with Codex, Codex is an iterative spatial proteomic technology where you first stain with this barcoded, a DNA oligo barcoded antibody, and you cross-link to the cell. So we can do roughly from 50 to 100 antibody or features. And then you anneal 3 oligos, which has fluorophore attached to it, and you image. From there, you denature that cross-link, and then you redo it again, and iteratively, that's how you achieve the multiplex and multiple antibody on the same tissue platform. So it's currently commercially available through the company of Koya. So it is really challenging to work with multiplex imaging, not just because the size ranging from, let's say, 400 MB to 20 gigabytes. It's also because you don't really know how to achieve these beautiful images at the first place. And more importantly, how do you extract information from these images into insights, or at least your manuscript figure 1, right? So the challenges between these two connections, first of all, is the availability of your favorite bioinformatician, right? I'm sure you have one, or you are the one, you have a lot of things lined up. So that will be a hurdle. Second of all, it's not quite intuitive. If you look in around on the field, there are quite a lot of packages, but how useful are they? You sort of have to test them out, and you, for a bioinformatician, probably want a robust, easy to use tool. So in today's talk, I'm going to introduce Spatial as a end-to-end platform, multi-layer, meaning we get single cell, cellular neighborhood, community, and tissue, and user-friendly. Right? So we are hoping to publish that or at least submit that soon. So first of all, you probably are using or know of some other existing packages. So why are we creating something new, or what are the differences? So let's take a look, right? So for analyzing spatial, at least codex images or MIME images, there are 4 major components. So the first one is the cell segmentation, right? So each images contains cells, and you wanted to know what are the markers that are contained in this cell of interest. Granted, you can also do cell segmentation-free analysis as well, depending on your purposes. But this is the common first step that you would do. The second one is the data pre-processing, which is often neglected, but a super important quality steps for your downstream analysis to be meaningful, right? So that includes artifact removal. That could be a particle dust from your kin wipe. It could be something that's super sticky, like your bone marrow leftovers, right? And then you do normalization, and that also we have a paper on that, on which is the best normalization schema. And also there are noisy cells, right? For whatever reason, some cells are just sticky and positive for everything, and you want them to remove. And from there, you also wanted to do the cell type annotation. If you're operating from a cell-centric sort of hypothesis, that could be using the super unsupervised clustering methods or sort of more machine learning-based approaches. And from there, once you have the cells, the X and Y, that's where the spatial analysis come in, where you can compute for cellular neighborhood analysis, understanding the niche and how they operate with each other, how that vary across, let's say, time or disease condition. So using these like 4 major like framework of what a package should contain, let's take a look at the existing package. So all of these item or roles, each one of them is a package. So I am listing out in column what are the functionality that they possess that are essential for the analysis of spatial data, particular codex, right? So as you can see with a yes and no, it's not necessarily consistent that you have all the feature in one package, and that's what we try to achieve. So that's feature number 1, that we can do all the analysis and all the steps in one package. The second thing that you want to take a look at when you're operating all these packages is that how many languages do I need to know, programming language that I need to know to operate from end to end, right? So I colored the programming language in different color, and you can see that you need to switch quite a bit, which probably is not a problem if you are coming from like a very computational background, but it's a hassle for me as well. But for a lot of user, particularly experimental collaborators that we have, we would like to provide an environment. You just need to at least have some minimal programming language in Python that would allow you to achieve from that to images to a figure 1. So the design principle so far right now is relatively easy to use. So we use a lot of Jupyter Notebook and Docker images, and you need a little bit programming language, but that is just to understand what code or parameters that are listed. And we have Docker environments so that you can even forgo the necessity of installation, which is also a pain for a lot of times, particularly some of the segmentation requires TensorFlow, which for those who work with it is not particularly easy and compatible. And we are using NData as a backbone. So for people who work with single-cell RNA-seq or some sort of data, and NData is an SC-verse compatible framework, which means that using this format, you can plug into an array of data packages that are available. And also we have interactive tissue annotation, right? A lot of times you want to look at specific regions, and the ability to draw that specific region that you're interested in is very critical for a lot of experimental design. And from there, once you sort of narrow down to what kind of parameters that you're interested in, we are hoping to scale that to a cloud computing sort of script, and that or allow you to batch all your examples and just processing in the same pipeline, but that is ongoing. So as I said, mentioned, so N-data is the backbone of our data storage unit where you have the protein feature here, and you will store all your metadata right here, and you can input all of like computation in the intermediate steps that you can put into it, right? Again, this is, I think your next speaker, Fabian Theis, they developed this SC-verse. There are an array of packages that you could use downstream, upstream, parallel of our pipeline. So again, I said this is a multi-layer information, right? Well, I'll show you with the example, how can you get from single cell information, cellular neighborhood information, community information, and to the tissue. And these are functionality that we have designed with, like sort of modularization of tools and functions that you could use. So just to briefly mention that we have the core function, right? Most of the codex data, if not all, and other spatial data format with this is you can always get information and from it. And from the auxiliary function, so that requires a little bit of additional design. For example, you might need a little bit more replica, a little bit more conditions. So not all of these auxiliary functions will be useful for your purpose, your hypothesis, but they are available. And for quality control and visualization, we have designed all these functions along the way of your analysis to make sure that it's doing what you expect it to do, right? So having that visual feedback is super important for spatial analysis, particularly for an experienced user. So in the first application that I'm going to show you is a human tonsil data, just 2 images on how we can use like spacey. So again, so let's start from the first one. So in the first notebook, you have 2 codex images. One of them is a tonsil, normal tonsil data you get, and the other one is tonsillitis image. From here, spacey provides you with 3 segmentation options. So mesmer, cell pulse, or cell seq. So you can do either membrane or cytoplasm or a combination of other markers to do the segmentation. And from here, in the first notebook, which is like, I think 2 or 3 functions, you will be able to do the segmentation and then visualize the segmentation mask on top of your original image and to see how that works. But I would say, if you have a very large image, I would highly recommend you to do that with a GPU on the server and whatnot. But for this images, which is 400 megabytes, this function performs under 2 minutes, which is pretty short. In notebook 2, this is where we have the removing of the artifacts. So you can remove the artifact based on the size, right? There are certain particles that are much smaller than what you expect the cell size to be. However, that is sort of based on empirical knowledge of your data set. So that you can set as like at the bottom 1%, however you will like that. And for normalization, there's an array of normalization that we're doing, and we're also thinking about including a new normalization. And we do have a small paper on the different normalization, how that affect codex data. So feel free to check it out. And after that, we're also removing noise. Right? What we're defining noise is that after Z normalization, for example, anything that's greater than 0, we are considering it positive for that particular signal. If you have 50 panels, for example, and it's positive for 49 of them, chances are this is not a cell that you're looking for or a functional cell. So we would like to remove them, right? You can go stringent, however you want in terms of percentage of cells that you want to call noisy. And then from there, in notebook 2 was that as an end data, and that's where you can plug in into different packages that are SC-verse compatible. So with that, we are starting the interesting part, which is the cell type annotation. So since it's the SC-verse compatible end data framework, you can use all the ScanPy packages, which is one of the popular one in Python. So it's a Seurat equivalent in Python. You can do clustering like Leiden, Louvain, and you can visualize that using cell type specific marker to annotate. So in that sense, you need quite a bit of domain knowledge on what markers you're using and what are they representing. And we would highly recommend that you're using cell type specific marker to cluster first, then adding on to the functional marker, right? Because we have, I think Codex is a little smaller panel, so that tend to give us a cleaner results. So again, once you have annotated cell type, you can project it with spacey, you can project it back to your original spatial coordinate. That can give you an idea of whether you are clustering correctly in an anatomical sense, right? So if you see these like a few big blob here, those are all germinal center B cells, exactly what you would expect in a tonsillitis case, where they form the germinal cell center, and that okay, my annotation makes sense, right? And you can also visualize them in sort of cell type proportion, and depending on the sample size, you can do statistic on it on whether a certain cell type is significant in a condition or not. And you can also visualize them sort of on specific subtype of the cells. So for example, this is a little bit blurry, but you can see that with the tonsillitis case, you have much more germinal centers and much fewer Treg as expected. So you sort of have these sort of metric to understand how well your analysis is capturing the biology that at least you would expect them to be, right? So that's notebook 3. And obviously, through a lot of painful clustering, it's an iterative process. By no means it's easy. I have to work with a lot of clinical fellows and experimental biologists on each of the case to understand each of the cluster. But as we know, it's an advancing field. There's a lot of cell type annotation tool. Stellar is one of the methods that we also developed together with Marius, who now is a professor at EPFL, where we are leveraging the known annotation of a previous existing data set and developing this graph neural network-based approach to annotate the unannotated data. And also at the same time, we can identify novel cell types or the cell type that have not previously seen or exist in your reference data. So this is the machine learning-based cell type annotation that we are trying to incorporate into our Spacey. But that is also something that you need to know, that you need to have a ground truth data set in order for this to work. So as I mentioned, that once you have the clustering or the machine learning enabled cell type annotation, that's technically not it when it comes to cell type annotation, at least in the codex sense, right? What people tend to know is that with these marker expression, I would like to see them in the original tissue space with all the markers that I have imaged to see whether that works or not. So we incorporate this open source software developed by a group in Sweden called TissueMap that allows you to interactively to see where your cell type is. So for example, this is just a Jupyter environment that you activate and load. So what this does is that, for example, you load your image in, right? And each one of these dots is your cell with the annotation that you want to look at. You can zoom in and out or selecting just one cell type to look at, right? And that can tell you the location. And from there, you can also load in or your original marker. So, and they have like a very nice compression algorithm so that even though your data is 20 gig, it will be pretty small and pretty easy to interact with, right? So with this one, you can also draw the annotation and then export it out as a CSV file or a JSON file so that you have regional-based annotation. That's also what we use. So I have shared with a lot of my experimental collaborator. I think the feedback of using this particular one is quite good. Okay. So the neighborhood analysis. So for people who are not familiar with the concept, so what neighborhood analysis does is that you basically are, in short, clustering cell type based vector. So let's say if you have a sliding window on this already annotated cluster, so it doesn't necessarily have to be cell type, right? It can be cluster 1, cluster 2, or you can even consider adding in marker expression. So in each of the windows, it's a composition of the percentage of what particular type of cell that you're have annotated or clustered. And from there, you do a clustering. And depending on the parameters of the nearest neighbor that you computed or you selected or the number of neighborhood you want to visualize, that gives you the neighborhood mapping, right? So that, in essence, what it's trying to do is trying to tell you what in your environment, those cells have a similar environment in terms of the cell type composition, right? And that sometimes inform the functionality of that particular region. So in our notebook, which is notebook number 4 right now, for that 2 samples that I show you, we have cluster, and I've determined that 6 is the optimal number neighborhood that we will select. And this gives you an enrichment of what cell type are particularly enriched in that particular neighborhood. And you project it back to your original tissue space, and this is what you see. And we were super excited Actually, my clinical fellow is really excited because we actually see biology here. So that's why I'm gonna zoom it out and to let you know what exactly do we see, right? So in reference, this is the consult data that you will see from anatomy textbook, right? So if you see here, these pink neighborhoods that we didn't like specifically annotate are the germinal center, right? And as you can see right here, even though I didn't tell you the region name, you would know this if you're a trained immunologist or tonsil biologist. You would know these are the germ center, meaning that this is the tonsillitis samples because they are training to, like the infection to form a germinal center, as opposed to this one is relatively immature and small, right? So that's the pink one. So from there, you can already annotate that to be the germinal center. The other interesting part is this olive green area, which from, well, this one is not well annotated, but this is the mental zone, right? So this is where, the dendritic cells are sitting on there and regulating where the immature B cells coming in and out to get the education that they need to form the germinal center. So that's another annotation that's corresponding to the known tonsil biology, right? So what is exciting and also information that you could potentially learn from these type of analysis is in this pink area, right? So the pink area sort of strapped around the two inside near the germinal center, but also outside of marginal zone. But you sort of know that actually, if you look at the dendritic cell population, it has a different composition because the function of the dendritic cell here is recruiting the immature B cell to come in. Whereas the one here is a different type of dendritic cell that are trying to prevent the mature B cell from leaving, right? But in our data set, we weren't able to differentiate the two dendritic cells because of the marker. But from here, using the spatial data and neighborhood analysis, that could potentially be a way you can separate the two dendritic cells without the marker present, right? So again, and there are other, sort of like known, tonsil biology that you can annotate with the cellular neighborhood analysis. And from there, that's your neighborhood analysis with corresponding knowledge. So once you have demonstrated that this is capturing known biology and things that you see are not known, could be novel, could be something interesting. So that's where the power of the cell neighborhood analysis. And then obviously, we can also talk about cell-cell interaction, right? So what that means in a spatial sense is that, let's say I have an image, right? And then there are the cells that's sitting here. What if I randomly permute the cell type label and then in, let's say, 1,000 times, and I compute the expected mean of those cell pairs in compare to my observed mean, where would that fall? So in that sense, you can sort of see maybe the two cells or two type of cells are closer in one condition, which is, let's say here, right? So this is a known biology. I show you that the dendritic germinal center is so close to each other, right? So you see the blue is the tonsillitis one and the red dot is the tonsil one. So this is compared to the randomly permuted background and they are comparing to each other. And indeed, the tonsillitis one are much closer between the DB cell and dendritic cell as we would expect. And feel free, then you can go through the entire list using significant threshold or fold change threshold to identify cell pairs that, that are interesting for further investigation.
Sorry, can I?
Yeah. Yep.
This plot, is the x-axis a distance function?
The x-axis is fold change.
In distance or in expression?
In distance.
When? Okay.
Yep. So the fold change in distance and the expression is not taken into account in this one, but we can imagine. Yeah, we can imagine taking that into consideration as well. So patch analysis. So that's something that's a novel and developed by my master student who are co-first author in this paper. So what is interesting sometimes is sort of at the interface of things, right? So you imagine the tumor and like a stroma interface where you see these like finger-like structure where they're invasive towards the tumor front. So that's sort of the concept that we are getting into, right? So these are the germinal center that I've told you so many times, and we treat them individually as a patch. So what we are interested in is, let's say this is the patch. What are the cells sitting at the edge, right? So those are they similar because they are, like coming from the surroundings of the same neighborhood, or are they different, right? So we basically, once we have patch, we do a clustering around the cells that are around it, and not counting the ones that are in the neighborhood, but also just the ones that are surrounding it. And then we do that for tonsil. And then you can count depending on the conditions, and also like what are the cells that are surrounding it. So there are biologies that you can, look into as to what are the interfaces between it. We also have ones that are looking not just like individual patches, but also like interfaces between neighborhoods. But due to the time limitation, I'm not going into today. But if that's something you're interested in, we can also talk offline, and I can show you how that works. So as I mentioned, this is the tissue level interaction where you can draw like a brain area, you draw a region that you want to analyze, maybe that has a better staining, that also work, right? And again, we are using the tissue map as an interactive part. You can use it within the Jupyter, or they also have like a GUI on your desktop. So hopefully by now, well, in a previous iteration, well, you can see that most of the figure in that figure 1 are doable within 4 notebooks, right? 4 or 5 notebooks. So I hope to show you that SPA Codex or Spacey provides an end-to-end, multi-layer, and user-friendly approach. Right? So the whole thing takes 30 an hour, but again, it's the demos data, right? It always works. So from here, I wanted to show you the capacity of the tool in a multiple array of different data sets that are of different origin and of different question, right? So this is a collaboration with Yan, who's sitting here. We are working with human brain. So the question prompt is really looking at the spatial molecular mechanism of major depression disorder, right? And particularly in human, which are super precious. And we wanted to see at different molecular level, what are the mechanisms that drives the difference. So Yan and collaborators have this beautiful samples of control samples as well as MDD, short for major depression disorder. And we did both DBC, which is the RNA-based assay, spatial RNA-based assay, and we have spatial ATAC, and we have Codex, which is protein, right? So DNA, RNA, and protein, and obviously I'm focusing on the Codex one. So first of all, I just want to say we have a smaller panel for identifying different neurons, vascular cells, and immune cells, etc. Obviously, we can expand upon it. But with this one, so far, like the marker works well. We examine and deciding which one that we want to proceed forward. And this is just a snippet of the Codex data demonstrating this marker is exactly marking what we want it to be. And just using Spacey, this is a few markers from the Codex panel. You can already see the hippocampus like patterning quite well. And if you do just simple, not simple, you just do cell type annotation, looking at the marker expression and projecting the cell annotation onto the original spatial coordinate, you can already see, we can pick up some of the very interesting known biological and anatomic structure. And the second point that I wanted to mention is that Spacey scales very well with sample size. So we are actually currently trying to implement this like GUI GPU-enabled clustering, where we can cluster millions of cells in less than 2 minutes, right? Which is pretty good. Otherwise, I think for this one, I run for quite a long time. So we have 1.6 million cells right here. And as you can see that because the beautiful job that Graham has done, which is another co-author on that paper, we don't have much a batch effect, and so we can proceed with the spacing. Again, this is just to demonstrate that we see the cellular neighborhood that are consistent with anatomy. So if I'm not telling you these are the projection of neighborhoods onto the brain structure, and if you refer that back to the manual annotation of the brain region, you can see the striking similarity between the two. Right? So these again is unsupervised annotation of brain region. So that's example number 2. So Spacey works well, bless you, works well with both human tonsil and human brain data, right? A very different panel. Now I'm going to show you a slightly different direction of the project. Still working with Codex, but this is touched upon my interest in 3D biology as well. So we are interested in collaborating with Dr. Hendrik Messel, who's in the Netherlands. We are interested in the initiation of DCIS, which is short for ductal in situ carcinoma. So you can appreciate this 3D clear human mammary gland, which is like in section. So the scale is in the millimeter, right? We section it. Sorry about the lighting, but it's normally a very cool image. What we do is first section or first image them, the tissue cleared one.
And my only worry is to make the home go well.
Sorry, is there a question? Oh, someone's probably unmuted. So, so why do we want to look in 3D, right? So first of all, this is just a pre-menopause normal breast. And if you look at it, zooming in, so this is what you expect a normal or textbook-like a mammary gland to be like, right? It's a grape-like structure. But at the same time, if you see in the 3D images, they are not all the same, but this is, again, a healthy human breast where you have this hypobranch structure, the dilated structure, and the filled structure, right? And then if you look this across time and across people who are at risk of BRCA mutation, the composition changes in terms of what type of 3D structure that they have, right? So to arrive at that, of that huge 3D image, it's mostly manual work, right? It's a lot. So one of the directions is basically, can we develop like 3D segmentation algorithm to look at the structure differences? But at the same time, you want to know, because the tissue is so large, if we're looking at 3D, you can probably do 5 markers at the same time, right? That doesn't give you the microenvironment, the tissue tumor microenvironment that you want to look at. That could potentially correlate it with the changes in different structures. And maybe one of the environment is the reason to drive the progression of DCIS, right? So what we then do is develop this Codex marker panel specifically for human breast, which is more than 40, and we have tested. And now we are, again, normally is a very pretty image. We have successfully stained these pre-tissue clear images, and we are ramping up the number of sections that we are doing. And what the beautiful part is that I can trace back to the original 3D scan and know each one of the duct, what are their original tissue structure, right? What are their ductal structure, right? So, you know, like, again, we can classify them with the help of my collaborator who is an expert in breast biology. And so now we have the tissue structure, that the 3D ductal structure annotation, and then we have the cell type annotation from Codex. What then we can do is looking at the environment for the external ductal environment, as well as the intra environment, right? So these are the special things that the spatial data can provide you. So it's an ongoing project. I won't share too much into details. Hopefully it will come out sometime. Future direction. So I probably just show a lot of Codex data here, but coming from a single cell RNA-seq world, I've always been interested in sort of interplay between RNA and protein and other data modality, and trying to understand their interaction and understand if we can predict one from the other or integrate them, right? So this is the integration part across spatial omics direction is what I'm interested in. The second direction is in the 3D tissue biology. So this is a collaboration with a professor at SUHK, my home undergrad university. Then they have a startup called Immunos. So what they are doing is basically a 3D multiplex imaging where you can bypass the technical limitation of just 4 or 5 markers at the same time and still looking at the tissue microenvironment from here. And this is human brain, but we can imagine this to be applied to many different data and tissue type. So with that, I want to thank my collaborators, specifically Tim, who is the master student that we are co-authoring the Spacey project, who is very driven, innovative, and develop a lot of very novel approaches within the pipeline. And also, obviously, Gary for the funding and for the guidance, and there for the Brain Project, Ron, and Yan, and Graham, and others on them. project, and we have Hendrik, particularly for the 3D imaging on the breast project, and Huming for the 3D brain project. Many others who I might not mention here, and thank you so much for the invitation, and thank you for being here and listening to my talk. So I will welcome any questions.
Maybe I'll start from this one. What are your approaches for integrating the DBC with codecs and also spatial effect?
So right now we haven't really started that, but the idea is sort of like going with a graph approach, right? So basically we have similarity in terms of the tissue structure. So that's sort of what we are trying to do right now. But it's like
some alignment between?
So we haven't really aligned different samples yet, but I think we need to take a look at the morphology because I haven't really played around with the spatial ATAC data to see how similar, even though it's the same sample in terms of morphology. So that could be where we first align with the morphology, or we could just align based on the graph of, you know, the XYZ or like the cell line.
By graph, you mean in the spatial sense?
Yes. Yes.
Can you explain a bit more on how you perform the cell type annotation? For example, is the annotation only based on the like the marker, the measurement data, or do you take the position information into account already when you perform that?
So for the traditional clustering, the Leiden and Louvain, that sort of like prepackaged by the scan pipe data, we are only looking at the marker expression that you have profile. But for Stella, we're actually taking to the XY, the spatial position into consideration. And in the paper, we have shown that it has actually 10 to 15% improvement in terms of the accuracy. So, so depending on your approach.
Do you have a general recommendation when we should take the neighboring information into account? Because even though I can see why it would be helpful.
Yeah.
But I think that concern is when you have super heterogeneous environment, there might be like some boundaries of the different cell types that if you take the neighboring information into account, it might mislead the annotation.
That's a good point, but I sort of approach this from like a convenience point of view, right? So you probably, when you started the project, you have idea of like what kind of cell types or phenotype that you wanted to see. And if the easiest way, to my opinion, would be using clustering, arrives at that information and sort of and if you see there are information that's misleading because of the neighborhood that it's situated or the tissue that's situated, you can supplement it with a little bit manual sort of region annotation to augment that. If that works, I would highly recommend with that route. But if you know like all method fails, then you can try. I think because I think Stella is not particular. You need to have a very good annotation data set, which is also not something that's easy to come by.
Can you just explain? I was a little bit confused about like in 1H that like distance.
Sure.
Reading the labels.
So, of course.
Big picture.
Yeah, I was worried about time, so I cut this off. But looks like I was doing good on time on how to slide. Right. So this is like in the method. Wow. Right. So basically you can compute a distance based on XY, right? You just Euclidean distance. And so with the same Euclidean distance within the same sample, you just permute the label names, right? Like it used to be a B cell, then you can permute it however you want. And from there, you basically, so we sort of have a cutoff for interaction using 100 micron as a radius, right? So we consider that as interaction, but you can change that parameter however you want. And from each permutation, you can compute an average of the distance
between cell types.
So the interaction is based on types. And then you basically do a Wilcoxon test, and then the log4 changes is basically the distance. We have the expected and the observed mean.
So knowing that like the distance are either like farther apart or closer together, like how do you interpret that? And like when you're thinking about like what that means for your
Well, so I sort of refer back to the literature and trying to see in experimental senses or some sort of mouse experiment, have they observed this distance and what are the potential reasons for it, right? So with codecs, if you just you can design your panel to sort of look at like a receptor interaction, or you can use other data modalities such as RNA-DB or Visium to look at whether the distance is also coupled with the changes in terms of transcriptomic profile or sort of interaction profile. So I think that's another way for you to look at if this distance is signifying some sort of changes.
So is it like right to kind of think about like if the distance is much bigger between like after permuting and then
Yeah, it's less likely that, well, I would say that like from my perspective, the further away, the less likely they will interact, but also that's contingent upon like what kind of interaction modality you're talking about, right? But again, like the further away, even you're using paracrine signaling, the effect is much less, right? So, but we haven't really modeled upon like what that's signify, but this is intuitively how I would interpret it.
Mhm.
Yes.
I've heard with codecs, there's like an issue where sometimes proteins diffuse across cell membranes, like between the time where like you're entering it and very different passing on the proteins. So like it's possible that for some given cell segmentation mask, you actually get measurements from your neighboring cell. So have you like hit this problem before, and are there any like tools in your package that address it?
I'm not quite familiar with this protein diffusing problem. Maybe if you have a data set or like particular one that I can take a look at. So that I have not personally encountered, but then it's definitely possible. Like for example, if you have a cell that has like a very strong signal, right? For the segmentation, sometimes the neighboring cell could pick up the signal that's actually from the adjacent cell. So that is, I think, an inherent problem for all the segmentation problems, which unfortunately it's not what I'm going deep under. But I definitely know in terms of, for example, the cell post design of the segmentation, what they were trying to do is really trying to look at this bordering cell effect, right? Where you can look up the paper in terms of how it's designed. So I would recommend trying out different segmentation method and see if this is still giving you sort of the separation of the marker intensity as you would expect, but definitely a little bit overlap or like spillover is something that I think would be pretty common.
Well, now one question also, actually, two. Question we had as well.
Yeah.
The Spacey. Oh, okay. First of all, is Spacey available to use, and where can we find it?
Yes. It's on GitHub, and it's we have actually taught a workshop using Spacey, so it works quite well. But I'm trying to clean it up by the end of the month, so hopefully I'll make it public and soon, and within a month or two.
And second question, does Spacey address batch effect, and if so, how? General insights about extracting information by computational method development versus experimental technology development.
I will answer the first question.
Oh, yeah, maybe the first one. Just does it address batch effect?
So in current pipeline, we haven't really incorporated batch effect workflow into it, but with ScanPy, its external package actually has a variety of batch correction method for your choices. And we have worked with using Harmony to batch correct some of our codex data, and works quite well so far. So I would recommend that to start with. But I think with batch effect, you could also try a little bit different approaches for that. And for the second question, could you reiterate?
Sure, yeah, I'll read. General insights about extracting information by computational method development versus experimental technology development.
Oh, that's a very
good question.
I think they're complementing each other, right? So a lot of computational insights that you have gained, you always need experimental technology to either validate or sort of to supporting its role. At the same time, if you don't have a great experimental technology, you won't see much computationally. So I think that's my insight. Probably I could probably answer that a little bit better.
One last question, and kind of related to a question I had too. So we're seeing in our book, like different tissues, marrow, breast, the choice of the first steps, the filtering, the normalization, really impacts all the downstream annotation, neighborhood definition, all of that. And if you're looking at complex samples like tumor tissues, it's hard. You don't really have a good benchmark to visually see your or quantify expected number of changes in the cell type, for example. How would you go about selecting these parameters?
What
parameters? Filtering and normalizing.
Oh, I see. I think I have to say the honest answer is empirically, right? We definitely have some default recommendation that we recommend people to do, right? Like we try Z normalization. And then for the filtering, we go quite stringent in the first pass. And then
what is stringent?
So for us, like for my we do like 5 to 10%.
Like the top half of intensity.
Yeah, so we do that to start with. And then in terms of clustering, like how do we know it worked or not, right? So basically we try to figure out, you first annotate, right? There's different type of tumors, and there is, immune cells. And we use the tissue map as an interactive way to look at the original tissue space and to see whether it makes sense or not. So this whole process is rather iteratively and empirical, and I definitely recommend having the domain biology expert in the loop with help with that. Has been how I approach it so far.
Kind of like building off that, like even before that, when you first get your data and you're looking at your actual markers, what are some like checks you're looking at to see like marker quality of like these markers actually work and then these markers are terrible and don't? Like how are you judging that? Or is it just by eye?
Well, yes. Well, yes, for sure. So the first pass is that does it fluorescent everywhere, right? And when you get a DNA mark or the antibody, right? It has expected expression on where it should be, right? So first you check to see whether it's at expected location. Second of all, you look at the homogeneity of that expression across, right? We definitely have seen samples where this one has a lot more, the other has a lot less.
What do you mean by like expected? Based on like the histology, you like kind of have an idea of where cell types are, or do you mean by like
when you buy the antibody, if you go to like the sample sheets, it tells you whether it should be located at the nucleus, cytoplasm, which one,
location?
Location of the marker, right? And then so that's like definitely what you would look for. And to start with, what else? Yeah, and also like whether it's like homogeneously distributed in terms of your samples. So that's another thing that we look for. I think that's like the two
like big things.
Big things I would look for.
I have a question about the cell annotation. So I don't work with spatial, but I work with single cell RNA-seq and a type-seq, and this is like my main focus to do the cell annotation, right? So in your pipeline, first of all, you use the package that is similar to CRET, and then you use Stellar, and then you use tissue map to make sure everything makes sense biology.
So it's not like a sequential approach. So I would say Stella and the clustering are sort of like a parallel, like you can do one or the other, or you can do both.
Okay.
So tissue map is this visual interactive way for you to validate or sort of to look at your annotation, because in our experience, I think we do definitely see there are cells that are marked with a certain type that makes sense from the marker expression level. But when you look at the tissue original space, like one of the students just mentioned, it's because that's spilled over.
Yeah.
Right. So that's the sort of the flow.
Okay. And when you say that you need like previous annotation for using Stellar, what exactly you mean by that? Like you need something that is very specific for that tissue type from Spatial, or can you incorporate something from single cell RNA-seq or stuff like that into that?
So so for Stella, what we meant, at least in the paper, we are using human intestine to map a human intestine sample. So it's within the tissue. But in my PhD, I also did a cell type annotation machine learning based approach. I've definitely tried, as long as the cell type are shared, the tissue where it comes from, particularly for immune cell, it doesn't matter that much. So that's something that you can definitely give a hack and try to see how that works.
Sure.
Yeah. Did I answer your question?
Yeah, that makes sense.
Yeah.
Thank you.
Other questions? All right, let's thank you again.